Hi guys, today we will be looking at the revision on the topic protein synthesis. Before we get into the actual process of protein synthesis, we need to make sure that we understand the key points associated with that topic. So we will need to recap on the DNA. Of course, DNA, mRNA, tRNA, those are examples of the nucleic acids that are necessary to understand the topic of protein synthesis. So, DNA, of course, it's a double helix. And you've learned about the DNA replication previously, and you've known that both of the strands act as a template in the process of DNA replication. However, what will happen in protein synthesis, only one strand acts as a template. The key information about mRNA is then that it's a single-stranded nucleic acid. The differences between DNA and RNA are mainly found in the bases. So in DNA, we've got the bases adenine, thymine, guanine, and cytosine. Then in RNA, the basis stands as adenine, uracil, guanine, and cytosine. Though another difference in the nucleotide is the fact that in DNA we've got deoxyribose sugar, which is pentose. That means it has 5 carbons. In RNA, then we've got ribose sugar, which is example of pentose as well, with 5 carbons. Another type of the nucleic acid which is important in the protein synthesis is tRNA. tRNA is the single-stranded molecule, nucleic acid, but what's important here about tRNA, it's the fact that it has the amino acid binding site, and it has the bases the same as in mRNA, so adenine, uracil, guanine, and cytosine. Another important thing about the tRNA is the fact that it has a triplet. So a triplet, it's a sequence of 3 bases called anticodon. Anticodon is present in tRNA, and those 3 bases we will locate over there. So it's a sequence of 3 bases. Another triplet, which has a specific name, codon, so a sequence of 3 bases, we can then find on mRNA. Okay, so those are the key terms that you need to be aware of when you're learning about the protein synthesis. So the structure of the nucleic acids, DNA, mRNA, tRNA, and of course the function and the role of them in the process of protein synthesis. So triplet, one more time, there are 3 bases. Triplet on mRNA, it's called codon. Triplet on tRNA, it's called anticodons. Where we can locate then really important information of the codons, it's a codon table. You probably came across codon table many times, and this is how the codon table looks like. Codon table, as the term stands for, will have the triplets on mRNA, and this is really important information. So when it comes to past paper questions and you are asked here to work out the name of the amino acids coded by mRNA or DNA, depends of which sequences you are given, you need to use the codon table. So we're going to have a quick practice here. I wrote down the sequences of the triplets on the DNA. So we've got guanine, and adenine. We want to now make a complementary codon. Remember, the bases in DNA were complementary, where adenine was always pairing with thymine and guanine with cytosine. So those were the base pairs in DNA. However, in RNA, the scenario looks different. Adenine binds with uracil and guanine pairs with cytosine. So remember, that's the key thing. So if on the sequence of DNA I can locate guanine, adenine, then the codon on mRNA will be cytosine, and uracil. So we're starting with the DNA. That's the complementary codon then on mRNA. If I want to then find the complementary anticodon to the codon on mRNA, so remember that's codon, that's anticodon, then I'm going again according to the base pair rules. So cytosine will then pair with guanine and uracil with adenine. Now I've got 3 sequences on DNA, mRNA and tRNA. I want to find out what is the amino acid coded by those sequences. And remember, to find out the amino acid from the codon table, I need to follow the sequence on mRNA. So I'm trying to find CCU here. How to use the codon table? We're always going to have the first letter over there, which is the cytosine, C. Second letter, it's also C. And the third letter, it's U. So easily now I can locate my first amino acid coded by this sequence, which is proline. And one more time we will do that practice. So the sequence on a DNA, I've got adenine, guanine and thymine. I'm pairing those, so adenine goes with uracil, guanine with cytosine and thymine with adenine. I'll write down this now on the sequence of anticodon. So I've got adenine, guanine and uracil. I want to find out the amino acid coded by those sequences. Remember, I need to use the sequence from mRNA to access the codon table. So my second amino acid, it's coded by uracil for the first letter, cytosine for the second letter, and adenine for the third letter. So UCA I can locate here in my codon table, it's serine. Okay. Another important thing that you can use using the codon table, it's the idea of degenerate genetic code. Why is it important? It's the fact, as you can see in the codon table, that the same amino acid can be coded by more than one triplet, and more than one codon. So example here I've highlighted, it's arginine. This amino acid can be coded by following codons: CGU, CGC, CGA, CGG. If I look further throughout my table, I can also find this amino acid here, and those are further triplets coding for it. So I've got AGA and AGG. So one more time, degenerate genetic code stands for the fact that one amino acid can be coded by more than one triplet codon. All this information is necessary to be able to understand and apply yourselves into the questions based on the protein synthesis Also, can refer those to the mutation topic, where you talk about the effects of the mutation or no effects at all because of the degenerate genetic code, which will not change the amino acid. So what we're going to do now, we're going to go through the process of protein synthesis, because now you've got all the key terminology and skills required to access this topic. So hopefully you remember the process of protein synthesis differs in eukaryotic and prokaryotic cell. In the eukaryotic cell, we will have three main parts of the protein synthesis. We will start with transcription. We will also have splicing. And we will have translation. In the prokaryotic cell, we will only see two of them, transcription and translation. Splicing does not happen in the prokaryotic cell. The reason behind that is obviously because of the lack of introns. So if you look at the sequence in DNA we could have the coding sequences called exons, but also we would have non-coding sequences called introns. They don't code for any amino acids. So introns are present in the eukaryotic cell. However, we will not find them in prokaryotic cells. So as you can predict now, splicing is the removal of introns. Hence, in the eukaryotic cell, we will have the extra process called splicing, which is removal of introns in pre-mRNA sequences that obviously does not code for the desired amino acid. So we will start with the processes of transcription. First process of transcription happens in the nucleus of the cell. Process of transcription begins with the DNA. We did say at the beginning of our video that in the protein synthesis, in transcription, only one strand of DNA will act as a template. Similarly, like you've learned in the process of DNA replication, the hydrogen bonds between the complementary bases on DNA are broken thanks to the enzyme DNA helicase. So the role of DNA helicase is to obviously break hydrogen bonds and form DNA template, which is only one strand. Okay, so what I'm going to do now, I'm going to write just some bases on the DNA sequence. So far we've used DNA helicase, which breaks hydrogen bonds, allows the formation of the DNA template. Remember, only one strand of DNA acts as a template. Having this template exposed, the 3 nucleotides, but this time the RNA nucleotides, are going to align themselves in the complementary manner to the template provided by DNA. So RNA nucleotides now are attracted to exposed bases. But remember, the rule is slightly different. Adenine pairs with uracil, guanine with cytosine, cytosine with guanine, and thymine with adenine. So in this process, imagine if I'm working with the prokaryotic cell only, I've already produced my mRNA. However, if I was to work with the eukaryotic cell, now I need to do splicing. So I am not achieving straight away mRNA. With those bases here, I'm achieving pre-mRNA. And remember, some of the bases, so I'm just going to come up with different bases as the example. Some of those bases are not going to be coding for the protein. So because there are non-coding sequences, there are introns here. They've used the sequences of introns on DNA to obviously appear in our pre-mRNA. So for instance, those here are not going to be useful in the process. So I'm going to splice them out. I'm going to remove them. By removing them, as you can see, what happens to the pre-mRNA? Pre-mRNA losing those bases because there were introns on DNA, and the mRNA become shorter. So that's the process of splicing only in the eukaryotic cell. Okay, so what we've achieved here as the process of transcription and even splicing after, in each of them we've achieved our mRNA. The process of transcription took place in the nucleus. So if you remember the structure of the nucleus, it's enclosed in the double membrane, which we call the nuclear envelope, and that form, mRNA, now needs to leave the nucleus. It's going to leave the nucleus through the nuclear pores. So nuclear pores allow this mRNA to leave and enter the cytoplasm. In the cytoplasm, we've got the organelle called ribosome, which will allow the second process of the protein synthesis, translation, to take place. So now we're going to have a look how translation is taking place on the ribosomes. So we've achieved mRNA in the process of transcription. Or following splicing. This mRNA attaches to ribosomes. Okay, so I'm going to here draw a large ribosome. So this is the ribosome. Okay, let's just come back to the bases that we had on mRNA. Just going to go of the ones we had there. So U, C, C, G, G, A. Remember the triplets, so the sequence of 3 bases on mRNA were called codons. At the beginning of the sessions, we say that codons are complementary to the anticodons on tRNA. So let's remind you quickly this was the structure of tRNA, which has anticodons, so the triplet on tRNA, and he also has the amino acid binding site. So the job of the tRNA in process of translation is to, of course, bring amino acids. So what happens now? Obviously, process is happening on the ribosomes, mRNA attached to the ribosomes. What else comes along here? It's the tRNA. So tRNA comes along with the complementary anticodons. So remember, uracil binds with adenine, cytosine with guanine. Each process of the translation has to start with the start codon. You can read the start codon again from the codon table. So, mRNA attached to the ribosomes. Remember, the codons are found on mRNA and anticodons are found on tRNA. And this is where mRNA codon now binds to specific tRNA anticodon. So we've seen that formation of the complementary bases. Another thing that we are aware of is the fact that, of course, tRNA now will bring a specific amino acid. How do I know which amino acid am I bringing? Obviously, from the codon tables from our previous exercise. So I'm looking back at the sequence of UCC. What's UCC? It's coding for. So UCC, remember, first letter, C and C, it's coding for serine. So that specific amino acid which is brought by this tRNA, it's serine. This process keeps repeating and repeating. So the sequence of the codons determines the further sequence of amino acids that will be found in this chain. So we will now quickly form our second amino acid. So the same here, the tRNA comes along with the complementary anticodons. So I've got now CCU. However, I want to know what's the name of my second amino acid. So I'm going off the codon sequence GGA, locating GGA in our table. So GGA. This is the glycine amino acid. And the process keeps repeating and repeating. So the sequence of codons determines order of amino acids. So determines the primary structure. Obviously, the amino acids are joined together through the condensation reaction. Which is the removal of water. The bond that is formed between those amino acids, of course, it's called a peptide bond. So I'm going to quickly recap on the structure of the amino acid once we're on this part. So this is a single amino acid with the central carbon, R group. We've got 20 different R groups, amine group, and the carboxyl group. So once we're joining those amino acids, so there will be another one. Sorry, I've made here a silly mistake looking at the time, of course. Okay, we're joining those amino acids together, and of course we say it's a condensation reaction, which is the removal of water. Where the water is lost from the OH group, from the carboxyl group, and hydrogen from the amino group. So if I want to join those amino acids together now, so I'm going to remove that. Okay, those are gone. I'm going to achieve the peptide bond from between the carbon of the carboxyl group and the nitrogen of the amino group. So this is the peptide bond. And that is the whole process of protein synthesis. There are a lot of things to remember here. The synoptic aspects of the structure of the amino acids, the role of the enzymes. And another thing that we need to recall, one more enzyme here in the process of transcription that allows the bases to be pair. It's the bases to be pair, of course, that's thanks to the hydrogen bonds. However, the whole nucleotides of mRNA are joined together here thanks to the enzyme RNA polymerase. RNA polymerase allows the formation of the phosphodiester bond. Okay, so one more time, transcription is the process that allows you the formation of mRNA from the DNA. It happens in the nucleus. DNA is going to unzip thanks to the enzyme DNA helicase, which has the function to break the hydrogen bonds and allow DNA to act as a template for the process of transcription. Remember, only one strand of DNA got the ability to do so. Complementary base pairs now are taking place between exposed DNA bases and the mRNA nucleotides, so with the manner where adenine binds with uracil and guanine with cytosine. The hydrogen bonds are reformed between the complementary bases, but what joins the mRNA is the RNA polymerase enzyme, which allows the formation of phosphodiester bonds between mRNA nucleotides. If the process was taking place in the eukaryotic cell, splicing is necessarily to happen, which is removal of introns, non-coding sequences. They are removed from pre-mRNA to form the final mRNA molecule, which you can see it's much shorter, has less nucleotides, less bases, as we can see on the diagram. The final mRNA is then going to leave the nucleus through the nuclear pores and go to the cytoplasm and specifically to the ribosomes. This is where the second process of protein synthesis, translation, is taking place. on the ribosomes. So mRNA attaches to the ribosomes. Remember, mRNA has codons which got the ability to bind to specific tRNA anticodons. tRNA also has the binding sites for the amino acids, hence brings in a specific amino acid. This process starts with the start codon and obviously will finish with a stop codon, which you can locate in the codon table as well. Stop codon allows the attachment of the polypeptide. So final polypeptide deattaches. How the polypeptide is formed? Of course, thanks to condensation reaction, which is the removal of water. To recap on the structure of the amino acid, we had the central carbon, R group, which we've got 20 of the R groups, hydrogen, amino group, and the carboxyl group. So originally here, carboxyl group also had the OH. However, the OH from the carboxyl group and the hydrogen of the amino group from the second amino acids were removed because of the condensation reaction, loss of water, to form a peptide bond. Peptide bond is formed between the carbon of the carboxyl group of the first amino acid and the nitrogen of the amino group of the second amino acid. So this process is determined by the sequence of codons, which determines then the order of amino acids, so the primary structure of the proteins. So remember, a lot of things to memorize here, a lot of skills to be able to apply into past paper questions. So again, that is only AO1. You need to now go away and do many past paper questions to get confident